There are several approaches now being tried that are published in the literature and undergoing animal studies and one or two even human clinical trials. In the long term, there are several groups all over the world looking at gene delivery as one potential approach. If you can target a vector into the tissue of interest, like cartilage, can you alter the way in which the cells in the cartilage are behaving, and perhaps they are upregulating enzymes that are attacking the extracellular matrix of the tissue. There are ways that you could genetically modify those. So that's under intense investigation. It might be a while, but there is already a study from a group at the Mayo Clinic involved with rheumatoid arthritis and with gene delivery. And the hope is that eventually this might migrate into the field of osteoarthritis. It's not clear how long that will be, but that's certainly an avenue that's exciting and under intense study. Currently, there are also approaches using microparticles, nanoparticles that might be delivered to the joint in the synovial fluid of the joint. And once they reside in that fluid space, they deliver a drug to the fluid. And once the drug is in the fluid, it can slowly diffuse, perhaps, to the target tissues. And that has been tried. Quite a few studies in animals are underway. And when you go to conferences in the osteoarthritis field, there are extremely interesting presentations on this. One of the potential problems with that approach is that whatever drug you might want to intra-articularly inject into a joint, like the knee joint or the hip joint, those drugs are rapidly cleared by the capillaries and the lymphatics that are in the synovium. So small molecules and even large molecules have a lifetime no greater than several hours in the joint. And so if you want to have long-term delivery to a target tissue inside the tissue, then it's incumbent to have extraordinarily high doses of that drug in the joint. And that might be one of the problems that occurs. So again, without a direct targeting inside the tissue, heavy or high doses of a drug in the synovial fluid of a joint may cause side effects that are not wanted. You need those high doses if that's the way you're going to deliver them, because cartilage has no blood supply. It has no nerve supply, and the tissue itself has no lymph supply. So cartilage lives by diffusion and convection. So when nutrients have to get to cartilage, let alone drugs, they diffuse into the cartilage from the synovial fluid. When you walk, of course, you're compressing the cartilage, deforming the cartilage in a complicated manner, and that compression and tension and shear causes fluid to move in and out of the tissue, and that might help transport nutrients and other molecules from the synovial fluid. But there's no blood supply, so if you want to get a drug into the cartilage immediately and have it reside there for a while to be able to act over weeks or even months, a different approach is needed. So one of the things we've been doing in our lab that our students have been working on, it was the work of a recent PhD student in our group, Ambika Bajpai, and she and other people in the lab had been looking at the use of small, highly charged nanoparticles, and it turns out that charge and size are critically important for an approach where you can introduce a nanoparticle into the joint space, into the synovial fluid, have that nanoparticle target immediately into cartilage tissue, and then deliver its drug inside the tissue. So the idea is to have a nanoparticle to which you can functionalize a particular drug of choice that you think might be efficacious, have the nanoparticle carry the drug into the cartilage, have that nanoparticle reside inside the cartilage for weeks, potentially months, and then deliver the drug inside the cartilage where you're not fighting clearance from the synovial fluid. So why is charge and size important? Well, it turns out that cartilage is such a dense tissue that particles less than about 10 nanometers are the only ones that are able to get in. Anything larger than about 10 nanometers or so just can't get in. In addition, cartilage is a heavily negatively charged tissue. It is composed of a dense extracellular matrix that has one of my most favorite molecules in all the world called aggrecan, a molecule that is critically important to cartilage function because it helps to resist compressive and shear loads, but it happens to be very negatively charged. So if you were to try to use a negatively charged nanoparticle, it might be able to sneak in but wouldn't get very far. But if you used a heavily positively charged nanoparticle, then the hope was that it might be able to be accelerated in, if you like, because of electrostatic interactions. So we've been using avidin as a natural protein that is found. It happens to be about 65,000 molecular weight, on the high side for anything getting into cartilage, but because it is so heavily positively charged, there is a partition in. If this is the cartilage and the bone and the synovial fluid, a positively charged particle will want to partition upwards into the tissue, and that gives a nice steep concentration gradient inside the tissue that helps to accentuate diffusion of that nanoparticle right into the tissue. So Ambika has done experiments in the lab along with other students working on this project where they have shown with cartilage explants in vitro that avidin functionalized to a drug. I'll describe the drug in a minute, but it's functionalized to a drug. It can carry the drug into cartilage in vitro in culture and then deliver the drug. And then if that cartilage is challenged by an inflammatory cytokine, which we used, which would normally occur in the joint space after ACL injury in a young person, we simulated that condition in the laboratory and found that the ability for avidin to carry in this particular drug happened to be dexamethasone, which is a glucocorticoid. It can suppress the cytokine challenge and maintain homeostasis in the cartilage over weeks at a time. So avidin turned out to be a really good example of a particle just the right size, having just the right charge to be able to carry a drug into the tissue and deliver it. Ambika then did animal studies, including a study with rats and a second study with rabbits, showing that avidin could penetrate rapidly into the cartilages and other tissues of the joint where you might want it, like the meniscus, and a particular tendon that happens to have a high aggrecan content. And right now there's an animal study underway in which the avidin carrying the same glucocorticoid, dexamethasone, is being tested in a well-used rabbit model of a traumatic joint injury, an ACL tear in the rabbit. So that's going on right now.